In this video, we're going to now talk about the processing of pre-messenger RNA. So we looked at transcription, we looked at the production of messenger RNA by the use of RNA polymerase, but remember that messenger RNA is not mature yet. It still needs to be modified in 3 different ways. So here is the gene within the DNA. It had the promoter, it had exons, introns, it had that polyadenylation signal sequence. And here is the product, the immediate product of transcription, where the introns are still in and that poly A signal sequence is still in there as well. Notice the promoter does not get transcribed. So this is a pre-messenger RNA, and there's 3 processing events that need to occur. Number 1, we need to add a 5' cap. That goes to the 5' end of the RNA. We need to add a poly A tail to the 3' part of the RNA. And we need to splice out these introns and splice together the exons. So the 5' cap is a modified GTP. So it's a modified G that is added backwards. So it's a 5' linkage instead of a 3' 5' linkage. And it's added to the 5' end of that transcript. Now, what is it for? Well, it protects the 5' end of the messenger RNA from degradation. Okay, there are all sorts of enzymes within the cell that will eat up RNA if it's not protected. It aids in the export of that mRNA from the nucleus, where it's being processed, to the cytoplasm, where it's going to bind to ribosomes, and then translation is going to occur using it as a template. And it helps that messenger RNA bind to those ribosomes. Now, on the other end of that RNA, we have the 3' poly A tail. And this is a series of A's, between 50 to 200 A's that are added after transcription is done. It's added to the 3' end. So this isn't something that's encoded within the gene. You won't see within the template strand of a gene T. There's a special enzyme that just adds a bunch of A's to the end of the 3' end of the RNA. Now, why do we need a poly A tail? Again, kind of the same reason as that 5' end. It protects the 3' end of RNA from degradation. It aids in the export of messenger RNA from the nucleus to the cytoplasm, and it helps the binding of mRNA to ribosomes. So basically, it serves all those same functions that the 5' cap does for the 5' end of the RNA. Now comes the most complex processing, which is called RNA splicing. And what RNA splicing is it's removing these introns, and these introns can be quite large. Removing the introns, and then splicing together each of the exons. And so what you end up with at the end of splicing is you end up with your mature messenger RNA that will then be exported from the nucleus into the cytoplasm. Now, the molecules responsible for splicing out those introns and splicing together those exons is called the spliceosome. And the spliceosome is made up of what are called small nuclear RNPs, or ribonucleoproteins. And we say SNRPs. Now, a SNRP, a ribonuclear protein, is made up of a small nuclear RNA and a protein. The two work together. And what's special here is that the RNA molecules within those proteins are complementary to the exon-intron borders. They recognize where that intron is and where the exons are. And they sort of guide that spliceosome to the introns and therefore catalyze the splicing out of those introns and the splicing together of the exons. So by the end of this, then what you have, after all of the modifications are done, you have this mature messenger RNA in which all of the introns have been removed, all of the exons are stuck together. The 5' end has the G cap, and the 3' end has the poly A tail. At that point, the RNA, the messenger RNA, is ready to move to the cytoplasm for translation, and that is the next video, the subject of the next video.